So first for some background information on the H. mephisto nematodes. They're a very unique type of nematode because they were found living in such deep subterranean environment where no other nematode has been discovered in. And here pictured at the bottom, you can see this was actually the gold mine they were found in South Africa called the Beatrix mine. And they were found living about 1.3 kilometers below the surface. So that deep below the surface, obviously very high pressure and high temperatures, up to 37 degrees Celsius, and also very low oxygen levels at only 0.5%. And just for reference, us humans need at least 20%. So that really emphasizes the point that they thrive under abiotic stress. And because these worms live in these environments, their body is also adapted to have an extremely slow metabolism in order to conserve its energy and resources. And so these worms are very microscopic, much smaller than C. elegans, only a few millimeters in length. But despite their small size, they actually have incredible adaptations that have allowed them to withstand such high temperatures. And so one component that scientists first found when discovering these organisms was a genetic component in which there was an increased concentration of heat-resistant genes, shown here specifically the heat shock protein. And essentially what this protein does is just ensure proper protein folding and prevent any aggregation from damaged proteins. And so this discovery was assumed to be the reason that they could withstand these high temperatures. Now, taking your attention to the right of the slide to observe their anatomy, you can see they are very slender and skinny. And this outer lining you see here is their cuticle, and it's an outer protective layer that helps to protect them from the environment and also maintain their structural integrity. And they also lack a spermatheca, so they don't have the receptacle that sperm would go in when mating, making them asexual organisms. And lastly, pictured in the middle here, their head, you can see their mouth is a very unique shape, and it's shaped that way because it's adapted to feed specifically on the organic matter they feed on is specifically subterranean bacteria, and that's their predominant food source. So having that shape of their mouth allows them to ration this food for many, like thousands of years. So we chose these worms to work with just because, I mean, obviously they're very unique in the environment and conditions they were found in, which is unlike any nematode we've heard about or seen. So we just thought that it made them an exciting subject for our study. Along with the fact that only one was discovered, and it was the first subterrestrial organism to have their whole genome sequenced. And we also decided to work with them because we thought that they posed a lot of real world relevance. So I believe that studying the H. mephistos can really help us understand how not only they, but other extremophiles adapt and thrive in the extreme environments, which in turn can help us better understand their evolutionary processes. There are also agricultural applications, and by understanding their temperature-dependent behavior, we can develop effective strategies for pest control. And there's also medical implications just by studying their age, stress response, and diseases that can transfer over to other similar organisms that might be found. And lastly, I think investigating these organisms have just overall potential for discovering diverse ranges of populations in environments we would have previously thought to not look for. So the fact that these were found in these conditions opens up the question to where else in the world and what are the other organisms that we can't even fathom, like we know nothing about, that could possibly exist. So because of all these unique characteristics H. mephisto has and the implications studying them have, specifically the environment it was found in, being able to withstand these harsh conditions. We wanted to focus on how they actually were able to withstand living in these conditions, since it wasn't that well understood. And also just to find out if they could live in extreme conditions that were the opposite of what they were found in. So our main question was, how do different temperature conditions affect the behavior of the nematode Helicephalibus mephisto? And we hypothesized that in extreme cold temperatures, we used 4 degrees Celsius and 20 degrees Celsius. We thought that they would slow down in their behavior and movement, and their metabolism would become so slow to a point that they would just die off. And then we hypothesized that at 37 degrees Celsius, since that's the temperature they were found in, that they would thrive under normal conditions. And for the very extreme high temperature of 45 degrees Celsius, we thought that they would enter dour stage in order to conserve their resources. But in order to make sure our results were valid, we decided to compare the results from H. mephisto to these temperatures with a strain of C. elegans called DVIS 19. And so this strain, basically they produce GFP, so green fluorescent protein, under oxidative stress. So now I'll hand it over to Steve to actually discuss the results of incubating the C. elegans at the different temperatures.